Hello, dears, and welcome to Al-Hussaini Virtual Lab Pathology Talks, Tips, and Practical Tips. Today, I'm going to share with you an interesting case of fibromatosis and how really to interpret beta-catenin, because I know it is sometimes difficult to interpret. Usually, the staining is not very clear. So let's start with the case. So this is a 30-year-old male patient who was diagnosed as a case of familial adenomatous polyposis FAP, status post-proctocolectomy, has abdominal wall mass. And by virtue of his known diagnosis, we would expect this to be fibromatosis. And indeed, it was the case. So we have really proliferation of spindle-shaped cells with the formation of abundant collagen, and usually the spindle cells do not really touch each other. They are in a way separated from one another. There might be some degree of minimal pleomorphism, but certainly not more than this, and very occasional, if ever present, mitotic activity. One important clue that I find really useful in supporting the diagnosis of fibromatosis against the rest of the differential diagnosis is that there tend to be a formation of edema, this white space, because of edema around vessels, in particular arterioles in fibromatosis. And try it. It's really a very useful small tip that can, in a way, support the diagnosis of fibromatosis. And again, the cells don't really come so close to each other. They tend to be separated, and then we have the collagen. And these tumors are usually focally positive with a smooth muscle actin. But really the diagnosis nowadays relies on the confirmation by beta-catenin. Now, beta-catenin is normally expressed in the cytoplasm and cell membranes of cells. So the presence of cytoplasmic positivity in the processes on the cell membrane is normal. This is not, we cannot read it as positive. What is really positive in terms of the mutant beta-catenin is the presence of a nuclear staining. So whenever you encounter beta-catenin in any differential diagnosis, for example, in medulloblastoma wind type, in the brain, in endometrial carcinoma, what we really mean by positive beta-catenin is the nuclear stain. Other stains are normal. The cytoplasmic and the cell membrane staining is normal. Now, how do we really interpret the nuclear stain? Because this is difficult. I know this is difficult because usually the cytoplasmic and membranous positivity might make interpreting nuclear stain really difficult. So first of all, the way we interpret it, we have to look for vessels, lymphocytes in which we know that the nuclei will be negative. And the negative nuclear stain with beta-catenin would stain blue. This is the hematoxylin, the background stain that we use in immunohistochemistry. So we know that our stain is working properly and is not just overstaining everything on the slide. So positive, negative internal control. We call this internal control, negative internal control. Normal endothelial cells, normal lymphocytes that do not stain with the beta-catenin nuclear beta-catenin are our negative internal control. And even if you look here, for example, into the vessel, the cytoplasm of the endothelial cell is positive because, as we've said, this is normal staining. Now, in comparison, these are the tumor cells. Where is the nucleus? Where is the blue color like the negative internal control? There isn't any. It's all brown, it's all chromogen, it's all positively staining. This is the way I interpret beta-catenin. Again, negative internal control. This is really the usual staining pattern. This is the nuclear staining that we interpret whenever we want to say that beta-catenin is positive. And the way I report it is positive in nuclear stain between brackets mutant. If there wasn't a stain, I would say a positive nuclear stain in the membrane and the cytoplasm between brackets, non-mutant, no nuclear stain, just to make it clear to other pathologists reading the pathology report or to our colleagues, physician colleagues. So I hope you find this tip useful in your daily practice. Thank you.